Hello all participants, welcome you to the practice of the effect of sanitizing agents on reducing microbial disinfestation in ready-to-eat vegetables. As you know at present, minimally processed or fresh cut produce, including fresh cut or ready-to-eat vegetable, is become most popular at right now because it's healthy, fresh, and very convenient to the consumer. But the big problem is the vegetables is very easily contaminated with harmful germs or foodborne pathogen during the stage after harvesting until the point of sales. If you would like to remove all germs by using only fresh water, it cannot be removed all because the germ can stick to the surface of the leaf and sometimes it gets inside to the leaf. So today I would like to introduce you the new sanitizing agents as we call acidified sodium chloride or ASCs. So today the practice we will compare the efficiencies of the treatment or the sanitizing agents to remove all germ in the vegetables. We have three treatments. The first one, control, we just use only water to remove the germs. The second, we use the traditional ways is chlorine or sodium hypochlorite. And the third one is the new alternative sanitizing agents is ASC. Chlorine or sodium hypochlorite is the widely used the sanitizing agents at the present. It recommends to use in the range between 50 to 200 ppm. It can be used in fruit, vegetable, or the processing food. And the new alternative sanitizing agents as ASC. It has been proved by US FDA that can be used in fruit, vegetable, and the processing food as well. It's recommended to use in the range between 500 to 1,200 ppm. And the effect, it can be reduce the microbial and also prolong the shelf life of vegetables, raw fruit and vegetables. This session, I'm going to introduce you how to prepare the chemical that we use in this practice. In this practice, we use two kinds of chemical. The first one is 200 ppm of sodium hypochlorite, and the second one we use 500 ppm of ASC or acidified sodium chloride. Let's get started at the first one. In case of the sodium hypochlorite, we use this chemical, and this sodium hypochlorite contains 6% available chlorine. After the calculation, we have to take this sodium hypochlorite about 17 milliliters and adjust to the volume 5 liters with the water. In case of the water, we can use distilled water or filtered water or drinking water and mix well. Okay. In case of you would like to prepare more than 5 liters, in case of you would like to prepare 10 liters, you can multiply by 2. So you have to take the sodium hypochlorite about 34 milliliters and adjust to the volume 10 liters with distilled water or potable water. And the second chemicals, how to prepare ASC. In this chemical is a little bit special because the ASC is the combination of citric acid and sodium chloride. So the first one you have to prepare 5% of citric acid and 1% of sodium chloride. After that, you have to mix it together. So for 500 ppm, we will use 250 milliliters of sodium chloride. Okay. 250 milliliters of sodium chloride, and then add the water. It's about 4,350 milliliters of water. Okay. And then you add citric acid for 400 milliliters. and then mix well. After that, the important step is you have to check the pH using litmus paper. The pH of this chemical should be in the range between 2.5 to 2.9. How to check the pH is very easy. You just use litmus paper and dip into the solution. And after that, you just compare the color. Okay. This color show the range between 2.5 and 2.9. So this solution is ready to use in this practice. The preparation of ready-to-eat vegetable salads production. We have 9 steps. In the step 1, we purchase the green oak from the farm. After the green oak arrives to the laboratory, we have to select the good quality green oak and we have to remove the defect one. Step 2. After remove the defect one, we got the best quality of the green oak, and then we have to separate the green oak into single leaf. And step 3, we have to remove excessive dirty matters. We have to remove the dirty matters by using potable water. And then step 4, we have to wash again in the cold water at 10 to 13 degrees Celsius for 5 minutes. Because in this step, we'll make the green oak to fresh and crispy. And then step 5, we cut the vegetable to the small piece to make the ready-to-eat vegetables. After cutting in step 6, you have to put in the treatment that I talked before. We have 3 treatments. In the treatment 1 is the control. The green oak will be immersed in the potable water. And the treatment 2, the green oak will be immersed in the sodium hypochlorite. And the treatment 3, it will be immersed in the ASC. This all treatment will be take about 5 minutes. After finishing treatment, we will go to the step 7. In step 7, you have to remove the residue of the chemicals by washing in the cold water at 10 to 13 degrees Celsius for twice. You have to wash it twice for remove the odor and remove all chlorines. And then after that, Step 8. You have to remove the excessive water by using the centrifugation. And then the final step, after you remove the water, it will be packed in the commercial plastic bag and stored at 10 degrees Celsius. In this practice, we will check the two parameters. In the first one, we check the appearance, the quality of the green oak. We will check the freshness and the surface appearance of the vegetables. And the second thing, we will check the microbiological tasting by using a total aerobic pet count. And we use the PCA agar. We count the colony on the plate PCA agar, and then we count the colony at the day 0 sample and day 5 samples. After we count the colony number will convert to the log number. And then after we get the data, we will compare the results between day 0 and day 5 to study to investigate the effect of the sanitizing of the appearance and microbial count. This is the result of the practice. We have 3 treatments. The first one is the control treatment. It means the green oak was immersed in the potable water for 5 minutes. And the second one is sodium hypochlorite treatment. The sample was immersed in the 200 ppm sodium hypochlorite for 5 minutes. And the last one is ASC treatment. The green oak was immersed in the 500 ppm of ASC treatment for 5 minutes. Okay. And if we see in the appearance and the freshness of the sample, the control treatment seems a little bit wilting when compared with these 2 treatments, okay, and show the browning color at the cutting surface of the green oak. And between the 2 treatments, sodium hypochlorite and ASC, we cannot see the significant difference in the freshness, okay. So, and the browning symptom is seem less than control, okay, as you can see. The control treatment show the browning symptom at the cutting surface when compared with these 2 treatments, ASC and sodium hypochlorite. So, the freshness seems the same between these 2 treatments, and the controls show a little bit wilting. Okay. This is the results of the Arabic plate count, okay, of the 3 treatments. We have 3 treatments, including the control. Control is mean the green oak immersed in the potable water. And this one is ASC treatment. And the last one is sodium hypochlorite treatment. Okay. In the upper line, this is the sampling of the colony on PCA agar at the initial day after the treatment before stored at 10 degrees Celsius. And the lower line is the sample that after keeping for 5 days. Okay, so you can see in case of the control, you can see a lot of colony on PCA agar observed in day 0, and it increased obviously at day 5. In contrast to these 2 treatments, ASC and sodium hypochlorite can reduce the contamination caused from the Arabic bacteria. You can see at day 0 and day 5, there are not significant different increase of the colony on PCA agar. Okay, and there are not significant difference between these 2 treatments. So what does this mean? It means that these 2 sanitizing agents has effective to reduce the microbial in green oak vegetables. During the production of ready-to-eat vegetables or salad, microbial contamination can be present on the leaf, on the apparatus, and in the knife. That can spread out to the production batch. So we have to solve this problem by using the sanitizing agent or 200 ppm of sodium hypochlorite. We will dip the apparatus or the knife into this solution for 5 minutes. After 5 minutes, they must be washed by using potable water or drinking water before uses to protect or prevent the spread out of microbial during the production.